chip followed by high throughput sequencing. It is almost same to the previous one, but the microarray step is changed, right? That's high throughput sequencing step. First step is same as that of the chip sequencing, identification of the bound base to additive transcription factors by immunoprecipitation. Second step is look here. The next here, the chip sequencing is same up till this step, immunoprecipitation step. So we treat, after this we treat, we bound them with a fluorescent tag and treat them with a microarray. We in chip sequencing, we directly sequence these segments through high throughput sequences. Okay, isolated single-stranded DNA fragments analyzed by high throughput DNA sequences. Oligonucleotide adapters are used for this process. They are added to the fragments and massive parallel frequencing is done. Here. So the step is the same. The second step is massive parallel sequencing. I will not explain the massive parallel sequencing in this process. Affinity measurements is the second procedure. First one was the measurement of binding preferences. We have find out the binding preferences by chip, second chip, exo, DNase 1 hypersensitive site sequencing methods. And the next is the second step. The second method is affinity measurements. Affinity measurements of the chosen transcription factors to many short sequences. It gives a quantitative description, sequence preferences, like what it is. If there are more than one transcription factors and there are more than one transcription factor binding sites, so transcription factor binding sites are the building blocks of the regulatory sequences, we know that. So these are present in variable numbers in different cells and in different genomes. So their binding preferences differs in each cell in different organisms. So there are different methods to test out that. First one is the PM base. It is a protein binding method. Different methods include PM base, Ashley Slack's myotomy, head flaps. So what is protein binding microarray? In this, a transcription factor of interest is expressed and then purified. We have purified that one, we will treat that with double standard DNA microarray. Yep, these microarray already have the different sequences of interest genome wide. So then we will wash them. Fluorescent antibodies added. The probe in the microarray commonly contain all possible 10 base pair potential binding sites. Yes, these are the microarray. We know what are the microarrays. They have the sequences, oligonucleotide sequences on them, about 10 base pairs. So each, and they span the specific region of the genome. So when we add a fluorescent antibody, a fluorescent signal is produced. We can locate by the signal which sequence is which sequence is that which is bound over transcription factor. I'm not following the transitions. Okay, second one is the high throughput sequencing. High throughput systematic evolution of ligands by exponential enrichment, that is HT slugs. A double stranded DNA mixture is incubated with immobilized DNA binding protein, washed, recover bound oligonucleotide, amplify, repeat, and then waited what? High throughput sequencing. Okay. This one is the incubation of the nucleotide pool with target molecules. Target molecules are the transcription factors of interest, and these are a nucleotide pool that spans a certain genomic sequence. And second is partitioning. What is the partitioning? Partitioning is, as we did in the chip and chip-seq, the separation of the bound segments with unbound segments, and then is the removal of unbound segments, and then is the illusion of bounded species, and then amplification of these one, and after amplification we do high throughput sequencing. And high throughput sequencing gives us an accurate data of each nucleotide sequence. It gives us a data in form of like A, G, T, C, one after other we do this.